So translation process starts when messenger RNA is produced. Hence, messenger RNA is necessary for the protein's makeup. Whenever you want to make the proteins, whenever cells want to synthesize the proteins, they need a messenger RNA. Messenger RNA is actually a copy produced by the DNA for the synthesis of the proteins, and messenger RNA contains a message or the codes are here that we have messenger RNA that contains the codons AUG, AAA, UGC, UGA, and finally UGA. So codons which are read by the ribosomes to produce or to synthesize the further proteins. So this is a this is a structure showing the general steps of the protein synthesis. So in order to synthesize the proteins, we have a need of certain machinery. We have the need of certain components inside the cell. Ribosomes transfer RNA and amino acyl transfer RNA synthetase forms the essential machinery for translation, uh, decoding messenger RNA into a polypeptide chain in the cytoplasm that work for the purpose of protein synthesis. We have the need of messenger RNA, and we have this copy, that is messenger RNA or mRNA, that contains two portions. One is the start codon, that is AUG, that starts from the 5 prime, and second is the stop codon, that is the UAG. The second thing needed is the ribosomes, and we have in the bacteria the ribosomes are complex molecular machines composed of ribosomal RNA and proteins. They consist of two subunits: a small subunit responsible for reading messenger RNA, and a large subunit that catalyzes the peptide bond formation between two amino acids. So, as I already told you, in the bacteria we have a ribosomes that has two subunits in the ribosomes. They have two subunits, a large subunit that is known as 50S, and a small subunit that is known as 30S. So a small subunit is responsible for reading the messenger RNA, and a large subunit is responsible for making a peptide bond between the amino acids. Ribosomes bind to messenger RNA and provides sites: A site, P site, and E site, where tRNAs dock and deliver their amino acids in sequence according to the messenger RNA codons. As the codons appear on the messenger RNA, and such type of anticodons will be brought by the transfer RNAs, and they will make a peptide bond, and in the final that peptide bond shall be released and a protein synthesis process shall be completed. And third thing, that is transfer RNA with the amino acids, that is the transfer RNA, tRNAs. tRNAs are the adapter molecules that recognize specific messenger RNA codes via their anticodon region and bring the corresponding amino acids to the ribosomes. So tRNA has the ability, or they have the anticodon portion that will be recognized by the tRNAs and bind with those messenger RNA codons at the messenger RNA on a small ribosomal units, and also they will bring the amino acids as of that codons. So these are the basic things, or basic points, or basic components required for the protein synthesis. So the first step in the protein synthesis: initiation. The transitional complexes forms, and tRNA brings first amino acid in the polypeptide chain to bind to the start codon on messenger RNA. To initiate the process of the protein synthesis, it is necessary that a transfer RNA containing start codon is read. The ribosomes scan the messenger RNA and finds the start codon AUG. This codon tells the ribosomes start making the proteins. Here, AUG codes for the amino acids. AUG codes for the amino acid methionine, first amino acid in the most proteins. The ribosomes initiates translation by scanning the messenger RNA from its 5' end until it encounters a start codon, which is AUG. During this scanning process, the ribosomal subunits, along with the initiation factors, inspect the messenger RNA base by base to locate the AUG codon in an optimal sequence context. When the ribosome finds this codon, the methionine initiator tRNA pairs with the AUG codon at the ribosomal P site, triggering the transition from scanning to elongation of the polypeptide chain. This process ensures accurate and efficient translation initiation, vital for the protein synthesis in cells. So, simply we can say at first step, ribosomes bind with the messenger RNA. At second step, start codon that is AUG is read by the ribosomes, and after knowing the start codon, methionine initiator tRNA that was discussed earlier binds with the messenger RNA at the P site and initiate the process of the protein synthesis. So here the initiation of the process starts. The first amino acid should be bind with the start codon, means AUG, to initiate the process of the protein synthesis. Protein synthesis shall be started or get started when that AUG or start codon is read by the transfer RNA containing the amino acid. Then in the next step, tRNA brings amino acids. The tRNA transfer RNA molecules bring amino acids to the ribosomes. Each tRNA has anticodon which matches the codon on the messenger RNA. A specific amino acid, on the other hand, transfer RNA molecules play a critical role in protein synthesis by acting as adapters that bring the specific amino acids to the ribosomes. Each tRNA has two features, an anticodon region and an amino acid attachment site. The anticodon is a set of three nucleotides that is specific to a codon on the messenger RNA. This allows the tRNA to match the codon sequence on the messenger RNA precisely. At the opposite end, the tRNA molecules carry the specific amino acids corresponding to the codon. In the first step we discussed. Now in the second step, the transfer RNA shall come and bind at the A site, which is beside the P site, and that transfer RNA has two portions. One part is the part of the anticodon, which has the codons on the messenger RNA, and second part is the amino acids. That amino acids are of that codon type. So first it will read that anticodons, and then it will give the amino acid to a polypeptide chain. During translation, the ribosomes facilitates the binding of the pre-RNA anticodon to the messenger RNA codon, ensuring that amino acid it carries is added to the correct order to the growing polypeptide chain. This process is essential for the accurate decoding of the genetic message into functional protein. The amino acids are linked together by peptide bonds, forming the protein's primary structure. So, what happens in the second step? tRNA will bring the amino acid along with that, and after reading the code, that amino acid shall make a peptide bond with the already growing polypeptide chain, and in this result, there will be the formation of the protein. Now, in the third step, amino acids are joined. The ribosomes moves along the messenger RNA 3 bases one codon at a time. For each codon, a matching tRNA binds. The ribosomes joins the new amino acids to the growing polypeptide chain. The ribosomes moves along the messenger RNA in the step of exactly 3 bases or one codon at a time during translation. For each codon read, a tRNA with a complementary anticodon binds to the ribosomes at the messenger RNA codon site. This tRNA brings the specific amino acids corresponding to that codon. The ribosomes then catalyze the formation of a peptide bond, joining the new amino acids to the growing polypeptide chain. After this, the ribosomes translocates forwarded by one codon, a movement facilitated by the elongation factors and the hydrolysis of the GTP for energy. This process repeats codon by codon until the entire protein is synthesized according to the messenger RNA sequence. So in this step or in the third step, we can say that first start codon was read, then next codons that were containing the amino acids were bound with the A sites. After binding, now there is a turn for the joining of the amino acids. So for the joining of amino acid, there shall be a peptide bond formation between the already existed polypeptide chain and the incoming amino acids. And after the peptide bond formation, there shall be translocation. Means the transfer RNAs shall be moved from one codon to another codon in order to elongate the process of the protein synthesis. Protein synthesis happens from codon to codon. First one codon is read and amino acid is developed, then that is translocated towards another codon, and that codon is read and the amino acid is developed, then it is further moved to the next codon, and this process of movement of the transfer RNA along with the messenger RNA is known as the translocation. So during the process of elongation, there is peptidile or peptide bond formation, and also there is the translocation. Then second step, that is elongation. In the elongation step, transfer RNAs brings amino acids one by one to aid to the polypeptide chain. Two types of transfer RNA molecules come for the synthesis of the proteins. One tRNA molecule that binds with the start codon, and a second tRNA molecule that will bring the anticodons that will bind with the codons on the messenger RNA, and they will be read. And after reading the codons, an amino acid shall be joined with the polypeptide chain, which will result in the protein synthesis. Step is the stop codon. When the ribosomes reach at a stop codon, UAA, UAG, or UGA, it knows the protein is complete. As there is a stop codon on the messenger RNA and that is read by the ribosomes, then the process of the protein synthesis shall be complete, and ribosomal units shall get separated due to the release of the release factors. When the ribosomes reach a stop codon on the messenger RNA, which can be one of three sequences UAA, UAG, or UGA, it signals that protein synthesis is complete. These stop codons do not code for any amino acid. Instead, they are recognized by the protein release factors rather than tRNAs. Upon recognition, these release factors bind to the ribosomal A sites where the stop codon is located, triggering the hydrolysis of the bond between the polypeptide chain and the tRNA in the P site. This reaction releases newly synthesized proteins from the ribosomes. Subsequently, the ribosomes disassemble into the subunits and release the messenger RNA, ready to be recycled. This termination process ensures that translation stops exactly at the end of the coding sequence, preventing the synthesis of the longer or faulty proteins. So, at this step, we can say that the process of the protein synthesis is complete, and stop codons are read by the ribosomal units, and release factors are the released that will cause the separation or that will separate the ribosomal subunits, and no further protein synthesis will occur. We can see the third step is termination. Release factors recognize stop codon. Here is the stop codon on the messenger RNA, that is the UAG. That is also read by the release factors. Release factors recognize stop codon, and translational complex dissociates, and complete polypeptide chain is released, and the process of the protein synthesis stop, and the components are recycled. So protein synthesis is completed in three steps: initiation step, elongation step, and termination step. Here you can say in the structure of the ribosomes, smaller subunits, as we say 30s subunits are the smaller ribosomal subunits of the prokaryotic cells. At the 30th subunits, messenger RNA is bound, and first code or initiation code, start codon of the messenger RNA is read by the transfer RNA that is containing UAC, and that has one amino acid attached with that. So the first tRNA comes and binds with the start codon at the messenger RNA on the small ribosomal units to initiate the process of the protein synthesis. And after this, in the elongation step, as at first step, that start codon was recognized and the protein synthesis was started. Here a second molecule has come. Second molecule of the transfer RNA has come, and that has bind with the next codon. At first, start codon was read, and the second tRNA comes at the A site. There are three sites on these ribosomes. One is the exit site, and where there is a start codon, that is the peptidal site or P site, and beside that start codon there is a A site. The function of A site is to welcome the incoming uh, transfer RNA that contains the amino acids. So here on the A site, there is there is a transfer RNA that is containing an amino acid, and that amino acid shall bound or shall make a peptide bond with the already existing peptide chain, and it will get elongated to make a longer polypeptide chain. And in the third step, at the A site, there is a stop codon UAG, which is recognized by the release factor, and the process of the protein synthesis shall be stopped, and polypeptide chain shall be released after making a complete polypeptide, and the whole process will stop here. Here you can also see in this structure the translation ribosomal units and the messenger RNA and transfer RNA. So we have small unit of the ribosomes and a large unit of the ribosomes, and small unit and large unit are acting as like a sandwich, are acting as like a zinger burger. So small ribosomal units identify the messenger RNA specific sequence of amino acids on the messenger RNA and bind here with the messenger RNA, and after binding to this, the large subunits also comes to near and they bind up and start the process of the protein synthesis. So here the first RNA, which is containing a polypeptide chain, has bound with the initiation complex or initiation factor or initiation point AUG. Then the second, we have a second transfer RNA, which is incoming RNA that contains the amino acids along with the anticodons. That they come and bind with that codons on the messenger RNA and help in the formation of amino acid with the and peptide bond. They bring the amino acid, and that amino acid will bind with the transfer RNA on the P site, and in this way a polypeptide chain shall be formed, and a process of the protein synthesis may be completed.